My name is Leo Wong, and I'm an associate professor of neurology at the University of Washington in Seattle. I do clinical research in FSHD, and I'm here to talk about the RESOLVE study, which is the clinical trial preparedness to solve barriers to drug development in FSHD. I am here to speak on behalf of the FSHD Clinical Trial Network that is headed by Dr. Jeffrey Statlin of Kansas University Medical Center and Dr. Rabi Tawil of the University of Rochester. What is the FSHD Clinical Trial Network? It is a consortium of 8 United States sites starting from the eastern seaboard of Kennedy Krieger Institute with Dr. Catherine Wagner, the Virginia Commonwealth University with Dr. Nicholas Johnson, the Ohio State University with Samantha LaRusso, University of Utah with Russell Butterfield, Perry Shea, and the University of California, Los Angeles, as well as us, the University of Washington in the Pacific Northwest. We also have 3 centers in Europe: Nijmegen in the Netherlands, Centre Hospitalier Universitaire de Nice in France, and Centro Clinico Nemo in Milan, Italy. What are the goals of this FSHD Clinical Trial Network? Our goal is to hasten therapeutic development. This includes both drugs and therapeutic approaches which may improve quality of life. A successful clinical trial depends on several factors, including access to and the ability to recruit patients, a precise understanding of the natural history of the disease and the major contributors to disease variability, and lastly, reliable outcome measures that are sensitive to changes in FSHD. The data that we collect will help researchers or pharmaceutical companies develop therapies to benefit people with FSHD. What is the funding sources of our study? This was initially started by the generous donation of private donor for 4 initial sites. It was expanded with funding from the MDA infrastructure funds and the FSHD Society bridge funding, as well as funding from Friends of FSH Research. The major funding was from a grant given to us by the National Institutes of Health. We also received funding from Fulcrum Therapeutics for a subpart of the study. Now, what is the RESOLVE study? Our overall aim is to hasten drug development for FSHD by validating new clinical outcome assessments and refining trial planning strategies. The study is over 18 months. After visit 1, there are visits at month 3, 12, and 18. The inclusion criteria includes patients who are FSHD1, age between 18 and 75, with symptomatic limb weakness, but able to walk 30 feet without the support of another person, including canes, walking sticks, and braces are allowed, but not a walker. Lastly, they should consistently take over-the-counter supplements. The exclusion criteria are heart or lung problems that preclude the safe testing of muscle function, as well as orthopedic conditions that preclude the safe testing of muscle function. Other exclusionary criteria include regular use of available muscle anabolic or catabolic agents, use of an experimental drug in an FSHD clinical trial within the past 30 days, as well as pregnancy. The schedule of events includes the following. At visit 1, there will be studies done over 2 days. In day 1 and day 2, we will measure functional outcome measures, as well as this new clinical outcome assessment called EIM, electric impedance myography, that we'll discuss in a little bit. Visit 2 occurs after 3 months, and we repeat the same measures. At month 12 and month 18, we also repeat the same measures to see how things change longitudinally. What will be done at these visits? There will be 7 health-related questionnaires. There will be physical assessments that test strength and motor function. There will be a physical exam, genetic testing at the first visit, a DEXA scan, and electric impedance myography. Once again, I wanted to emphasize that there are a total of 5 visits over 18 months. What is this clinical outcome assessment that we're interested in, and what is electric impedance myography? It is a non-invasive technique to measure muscle health. We are measuring the electrical impedance characteristic of muscles. What we're trying to measure is the opposition the muscle presents to a current when a voltage is applied. In muscle disease, a variety of factors can influence this, including muscle fiber atrophy and disorganization, the deposition of fat and connective tissues, and the presence of inflammation. EIM captures these changes in the tissue as a whole by measuring its impedance characteristics across multiple frequencies and at multiple angles relative to the major muscle fiber direction. Here you can see the EIM machine hooked up to a computer and applied to a muscle. I can attest to the fact that this is a painless procedure, having done it on myself. We apply a wet wipe with saline and then measure the impedance with this rubber appliance that is placed on your skin of 6 muscles on each side of the body. This should take no more than 20 minutes. Our recruitment target is 220. By January 2019, we have enrolled 70 patients in the United States and 37 in Europe, and we still need help. Some of the questions that we get are, how much will this cost me? There is no cost to participate in this study. Another question is, can you help me with travel expenses? And the answer is yes. Funds are available to aid with travel expenses. How do you get in touch with us? We would love your help if you qualify. And these are our main contacts through KU Medical Center and University of Rochester. Please contact Kylie, Katie, or Leanne if you are in those areas, or if you're in other areas and need to find a local center. If you are on the eastern side of the United States, here are the contact information for The Ohio State University, Kennedy Krieger Institute, and the Virginia Commonwealth University. If you're on the western side of the United States, here are the contact information for the research coordinators who can give you more information and tell you more about the research that we're doing. Finally, I want to thank you for listening to this short webcast. We appreciate any help that you can give us.